Magnetoencephalography, MEG, is a non-invasive imaging technique for investigating human brain activity. It allows the measurement of ongoing brain activity on a millisecond by millisecond basis, and it shows where in the brain the activity is produced. It is entirely non-invasive, silent, and with no applied magnetic fields, radiations, or injections of any kind. In this module, you will learn what MEG measures, how it compares to and complements other neuroimaging techniques which you may be familiar with, and begin to appreciate some of the challenges of performing MEG measurements. MEG tracks brain activity by measuring the electromagnetic fields generated by neurons. During brain activity, large groups of neurons are activated. Those neurons generate electric currents illustrated here with a red arrow and yellow loops. These electric currents generate both electric potential and magnetic fields. EEG measures the scalp surface for changes in electric potential between pairs of electrodes which are attached to the skin. MEG measures the changes in magnetic flux or the magnetic field passing through the sensors which are positioned outside of the head. How does MEG fit into neuroimaging? We can categorize neuroimaging into three different modalities based on how they look into the brain. Conventional imaging methods such as computerized X-ray tomography, or CT, and magnetic resonance imaging, or MRI, give information of the anatomical structure of the brain. Functional imaging methods such as positron emission tomography, or PET, functional magnetic resonance imaging, or fMRI, and near-infrared spectroscopy, or NIRS, provide information on blood flow, oxygen consumption, and metabolic activity in the brain. Magnetoencephalography and electroencephalography, or MEG and EEG, measure functional and information processing. Only these two methods are a direct measure of brain activity in real time. Here you can see a schematic comparison of the spatial and temporal resolutions of these various imaging methods. The spatial resolution of conventional imaging methods like CT and MRI is excellent, being able to resolve the structure of images at the submillimeter level. However, they have a poor temporal resolution, often in the range of seconds. Functional imaging methods like fMRI, PET, and SPECT have lower spatial resolution, but can reach temporal resolutions of about 100 milliseconds. MEG and EEG, in comparison, have excellent temporal resolution, shown here to be sub-millisecond. However, their spatial resolution is not as good as some neuroimaging techniques. The spatial resolution shown here represents the accuracy at which MEG and EEG can localize brain activity. We refer to this as source localization. MEG is considered to have better source localization than EEG because the brain, skull, and scalp distort the EEG signals, while the MEG signals pass much less distorted to the outside of the head. We will discuss this in greater detail during the course. Let's compare the MEG and fMRI. MEG detects electromagnetic fields generated by neural currents. This means MEG is detecting the brain activity as it happens, recording the activity as fast as 1 millisecond or less. fMRI measures the hemodynamics, which are modulated by neural activity. This means that fMRI detects changes in blood flow and oxygenation, which takes longer to occur than neuronal activity, limiting the temporal resolution to greater than hundreds of milliseconds or even seconds. On the other hand, fMRI yields better spatial resolution of the anatomical structures. So while it may be possible to only image slower processes, fMRI localizes those processes with more accuracy. Faster processing can be imaged with MEG, but generally with slightly poorer spatial resolution than in MRI and fMRI. It is important to also note that signal-to-noise ratio of raw data, or the ability to detect a signal out of the noise, is comparable between MEG and fMRI. Most of the neuroimaging methods we discussed are invasive to some degree. In fMRI, the body is exposed to strong magnetic fields. In PET and SPECT, radioactive tracers are injected into the body, thus exposing patients to radiation. In intracranial EEG, such as electrocorticography and stereo EEG, the electrodes are implanted directly into the brain tissue. In contrast, MEG and EEG do not require any external field or radiation or implanted electrodes. In summary, MEG is a direct, non-invasive measurement of neuronal activity with high temporal resolution and good spatial resolution. As we discussed, MEG reflects the changes in neural activity over time. Therefore, similar to EEG, it can be used to track responses to external stimuli, as in this example. The 30 millisecond peak response after somatosensory median nerve stimulation is indicated by a dashed blue line in the field versus time trace of one MEG sensor. Field values at this time from all sensors are combined and presented as a spatial map in the upper right picture. The blue and red contours indicate magnetic fields entering and exiting the head, respectively. One can also perform source modeling to localize the origin of the M30 response underlying this map pattern. The localization information can then be superimposed on the subject's MRI to see the anatomical structure. This combination of functional and anatomical information is often called magnetic source imaging, MSI, where neural current source localizations estimated from MEG data are superimposed on the anatomical MRI of the subject or patient. Because MEG signals are extremely weak, measuring them is only practical using a superconducting quantum interference device, or SQUID. While this sensor technology is currently used for MEG, other potentially promising sensor types have recently been introduced. The current implementation, known as a DC SQUID, detects changes in the magnetic flux of the field that passes through the sensor. The SQUID is controlled by suitable electronics for converting the magnetic flux changes into voltages which can be read by a computer. More information about SQUID sensors and how they have been employed in whole head systems will be discussed during this course. To eliminate or suppress the large magnetic fields in the environment, shielding methods have been implemented, including operating the MEG inside a magnetically shielded room. The MEG system has a large number of SQUID sensors, for example Tri-X Neo has 306 channels, optimized design combining magnetometer and planar gradiometer coils in the sensor array enables efficient interference reduction with passive technologies as well as with sophisticated software shielding methods such as signal space separation. The first MEG recording was published in 1968 by David Cohen at MIT, and then he reported the first MEG recording with a SQUID sensor in 1972, shown here. You can see his MEG recording of human occipital alpha rhythm, also known as our resting rhythm. The top trace shows the MEG recording when a magnetometer is placed close to the left occipital region during eyes open and eyes closed. By the end of 2019, the cumulative number of MEG publications was nearly 8,000. After the pioneering work by Cohen, the number of MEG publications remained low until the early 1990s, when the first commercial whole-head MEG systems became available. Thereafter, the number of MEG publications has increased steadily, proportionally to the number of available MEG systems. It has been estimated that during the first 2 decades of SQUIDs, between 1972 and 1992, slightly more than 1,000 SQUID sensors were employed in all biomagnetic applications. After the introduction of the first MEG systems covering the whole cortex around 1992, about 250 commercial MEG systems were installed. That resulted in over 50,000 squid sensors worldwide by the end of 2015. To summarize, MEG is direct, non-invasive measurement of neuronal activity with high temporal resolution and decent spatial resolution. With vibrant interaction between neuroscientists, clinicians, physicists, mathematicians, and engineers, the experimental approaches and analysis methods were developed to establish MEG as an important method to study healthy and diseased brains. MEG has become a part of the brain research system, where MRI provides structural information, PET and fMRI provide metabolic and blood flow data, and MEG and EEG provide millisecond scale data of signal processing. The manuals that come with your MEG system contain important information about using your MEG, including safety warnings. You must familiarize yourself with this material before operating your MEG.